Good afternoon, everybody. And today I will give you a brief introduction of the DNA encoded library and also the solution provided at the WuXi AppTec. So today the outline will be a very, very brief introduction to this technology and our development of this platform and some of our future perspectives of Dell. So for thousands of years, people have been looking for therapeutic compounds, and we started with herbal medicine screening, and then we think we have learned some knowledge, and then there comes the alchemy, and then we step back, said, we probably don't know a lot, and then we went back to high throughput screening. And personally, I think at some point we're going to arrive at the machine learning and AI-driven drug designs, but between now and then, I think the high throughput screening and also other screening methods will continue to provide solutions. And as this interesting quotation here, that most of the drugs are still developed by accident. So a lot of us are still working on how to make these accidents happen more faster or in a more predictive way. So Dell has evolved to become one of those technologies. And just briefly, this technology is based on combinatorial chemistry. And then by splitting and pool techniques, you can make millions to billions of compounds in one test vessel. Different to the traditional combinatorial chemistry, in this technology, each step you do the synthesis, you also include the DNA encode, so that every DNA tag will encode a specific small molecule. And in terms of selecting hits, you take it's very straightforward. You take the soluble protein and immobilize onto the solid surface, and then you incubate with the library, and then you wash away the non-binder and collect the binder. And that process takes about a week to finish. And then you use next-gen sequencing to sequence each of the DNA sequences, and then you can read out what is the small molecule that is responsible for binding to the target. So this technology actually allowed a unprecedented chemical diversity and easiness of the assay development and a very, very fast turnaround time. So this technology has been around for 15 years. At the beginning, it was relatively quiet, and then in the recent years, it became pretty popular. And in this slide, there's some examples of different companies using Dell technology. They achieved a very attractive molecule. At the bottom is one of the compounds that entered a clinical trial by GSK. And I believe there are many, many more of these examples that are not published or about to be published. So besides the traditional drug discovery, the Dell technology can also be used in many different ways. And if you imagine this technology is focused on finding affinity molecules, any affinity compound that you can think of can be discovered by this technology. And another advantage is it screens the molecule while they are in the conjugated state. So any small molecule you discover is readily conjugatable to other moieties. And then you can easily imagine the most recent PROTAC heat can be well supported by this technology. And also into the biologics, the specific purification reagent can be discovered by it. And then you can also think about drug delivery. Showing to the left of this slide is just another example that you can use the DNA encoded library to discover small molecule vehicles that can drive another drug moiety inside the cell. That is breaking the biological barrier. So the landscape of the Dell, as I also mentioned, the concept arrived very early, 1992. At that time, it was in the academic space, and in the last 15 years, with the help of the next-gen sequencing, and more companies have adapted to this technology, and there are also many service providers. Most of the major pharmaceutical companies has acquired access to the technology either by internal effort or by collaborating with the Dell service provider. Okay, so WuXi, we also believe that the DNA encoded library could serve as one of the initiating point of the drug discovery, and could be included in our service. And our vision is, we can provide a Dell platform for hit discovery, and then we offer library construction, library screening, and the next-gen sequencing informatics. And once the hit was identified, WuXi actually is capable of helping its client of driving the hit forward and then all the way into clinic with other capabilities that WuXi has already built. And WuXi also has a perfect ecosystem for Dell technology. As you can imagine, this is a very multidisciplinary technique that involves from chemistry to biology to informatics. And WuXi is very well equipped of growing this technology in-house. Okay, so far the WuXi's capability can be summarized by this slide. And we are capable of provide end-to-end support from the library design to the construction, screening, and hit confirmation. We have built a library collection of more than 200 libraries, and the total diversity is exceeding 80 billion, and they're all structurally diversified. And we have our own proprietary informatics system. And also we have started offering services. We have more than 20 projects since September last year, and within these projects, we have successfully identified nanomolar hits in different target classes. So now I will switch here to some of our visions of where will this technology go. And obviously, there are a lot of demand in the library construction side, and people always wonder, how do I cover more chemical space? How do I make my hits more drug-like? And what is the synthetic fidelity, given this is a combinatorial technology? And also, how do we predict the potency coming out of the selection? And how do we transfer the affinity to from affinity screening to translate the result into activity? And then we have established collaboration with academic collaborators to develop to unlock more chemistry reactions to enable us to further expand the chemical diversity. And we also started developing the activity-driven selection methods, and those are all happening in-house. And most importantly, the informatics, this is our vision that the entire field has been lacking, and the data interpretation methods are pretty primitive. This is where we invest the most into, and then we want to understand what limits the chemical diversity in this technology, and how do we interpret the data more efficiently, and also is the bulk selection data reusable? And if you do the screen against one target, how do you reuse that data for other projects? So that comes down to the fundamental challenge of the current Dell technology, because more and more talented chemists have been unlocking a lot of chemical probability of building this library, and now the collection of the library has exceeded 10 to the 11th molecule, and however the next gen sequencing, it becomes a limiting step, because you can only have 10 to the 8th readout in one sequencing effort. So as a result, as exemplified in this plot, If you look at your sample at very, very high sampling ratio, you can get a very fidel readout of almost every single compound. But if your sequencing depth is limited, this is what you get, the very, very sparse look of your data. And now if you look into every single compound, you will notice 90% of the compound will receive a result of a 0, and then most of the compound that has number will be one copy. So a lot of Dell practitioners will end up in this space and started calling hits based on very, very low copy count. And you are actually missing the very best one here because it just happened to be not covered by NGS sequencing. So hopefully you will understand, the challenge that was imposed on the Dell technology. That is a very good challenge because the technology always advances alternatively, and we are hoping the next generation of sequencing will help us ultimately solve the problem. But right now, this is also a good opportunity. Just like the genome projects at the beginning, the coverage is very low, and that actually inspired a lot of data scientists to work on the algorithms to figure out how do we look at the data more efficiently. I'm showing here is one of our efforts that takes very, very diluted data and apply some machine learning algorithm to it, and we can actually reconstruct the overall map of the original library. As you can see, it is not exactly the same as the previous plot. However, it is close enough. Okay, and most importantly, you get almost fidel read of every single compound in this library. Okay. But that is just the beginning. Like I said, it's a very primitive approach of try to reconstruct the image. But to do that, I think, as a community, we need to invest more in the informatics and figure out how to do the data cleanup and how to do the data clustering. And most importantly, I think the Dell practitioners will be open to data sharing and collaboration. And again, as the genome project evolves, the collaboration seems to be the key of advancing the technology. So to that end, WuXi is also launching another effort that is targeted at the academic researchers, and then we design a platform so that we will provide a part of the library as a free reagent to the academics, and then the academic users will run the screen. They return the sample to WuXi to analyze, and the key to this social experiment is if we can agree to perform the experiment before signing contract, and only when a good result comes out, and we will discuss terms. We are hoping this can be a game changer, also to boost the collaboration between industry and academia. So that will be all for today, and thank you for your attention. If you have any questions, please find me.